Okay, y'all. Thanks so much for joining. I show my screen now. Okay, so I've updated the course schedule a little bit with the YouTube from last time, last lecture, and also rather than spanning the next lab into 2 sessions, we'll actually do a review before the midterm, just to cover some of the terms. I know there's been some, thank you for sending feedback to me. I know there's been some confusion about the readings and what's going to be on the midterm, and I don't want you guys to fret. Actually, a lot of the figures that I used, especially for last time on in the lecture, we're pulling directly from the reading, and the readings are meant to give you more background. And I'm also realizing that, you know, there's a lack of textual explanation that was problematic with the last lab 1. And so what I'm trying to do now is just find the really good content with a lot of text and background understanding on this stuff in tutorial form, and then point you guys to those texts and those fuller descriptions. So we're going to be doing that more moving forward. And I also updated the lab 1 submission. So lab 1 is due on Wednesday, and there isn't much else to do other than just I peppered a few extra questions in the lab submission. But you can see in the end of lab 1D, there's a new section called lab 1 submission. So this is the last of the lab 1s, and at the bottom of that is the lab 1 submission. And all you're doing is going to submit a path to the lab on Babylon, or on Taylor. So, oops. So the let's go back to that. And the idea of submitting it on Taylor is just so I can also rerun things and check things. And also if you're missing something, see where it might not be spitting out, etc. And I broke out the lab materials based on each part that we did. And I'm asking that you consolidate those parts into a single R Markdown document so that I can evaluate it easily as opposed to referencing different files. Just want to look at one R Markdown document and see all the outputs. And then here's the grading rubric. So these are all the things that if you ran the labs, then you should be outputting. And here's the pointwise breakdown on what each of those things is worth. And there are questions in here that might not have been previously in the lab. So please be sure to answer these questions. And it's more just asking you to interpret your output and make sure that you understand things like, okay, we talked about these term plots, and you know, if it's above a 0, it contributes towards presence, and if it's below 0, it contributes towards absence. So I'm just asking you to kind of do some interpretation of those figures, and similarly with the Maxent term plots and some variable importance. But it's really just generating the outputs that you did for your critter, and just stating the obvious from those outputs. So hopefully that feels and looks fairly straightforward to submit. I wanted to also point out that in one of the previous lectures, when we originally switched over to Taylor, when was that? Let's see, is it this one? Yeah, when we originally switched over to Taylor, I was suggesting that you symbolically link the courses folder. And one of the advantages of that is that you can just take the course materials as I update them on Taylor, which I'll do right now, and pull from that R Markdown to get going. So I'll do that as a little example. So here I did this, I in the terminal. So let me just start fresh with this. Here, if I delete this, it's actually, let me just show you, this is not the folder itself. It's, if I do ls-l a, that's going to list everything, even the hidden stuff. And it will show me that this is actually a symbolic link. It's going to courses EDS 232. So I can say ls courses EDS 232. And you can see that those files there. Now I can remove this EDS 232. It's not going to delete the folder. I can still say ls courses EDS 232. It's still there. It just removed this, which is a symbolic link. So it's a Unixy way of being able to reference places easily. In this case, to be able to put it in your home folder. So if I recreated that, so I cd tilde into my home directory, and then another command, so I add a little semicolon. So this is on the terminal shell. And then I link symbolically this courses folder, and then in this case in here to my home folder. And if I cd into EDS 232, and I look in there, this is the EDS 232 ML, it's the GitHub. This is this repository in there. And let me just do a git pull to get the latest. Okay, so you all could simply, once you do that, you could cd into here, open this guy, and then here we've got lab 2b for community ordination. So these are the R Markdown files used to create the website. And you could just simply open this. Now you would not be able to edit here, and you wouldn't want to. That's the course materials that are up on the website. But I could go File, open this, and then File, Save As, and then put in my home folder. Maybe I have my own EDS 232, and I save here. So the point of all that was just simply to give you direct access to the course materials in the raw R Markdown format, and you could actually just go from there. Now, if things update later, you have to manually do that, or rerun it, or recopy it, but and merge those differences. But that's the idea there. Yeah. So we're going to talk about ordination today, and sort of this idea. It's this broader picture of communities, and we're using unsupervised learning techniques, meaning we don't have like a response term that we're fitting. And then we're going to do reserves on Wednesday. This will be a small lab, fairly straightforward to do, where we're building up to going from predicting the presence or absence of an individual species based on the environment, to then looking at how different sites might vary across species, to then going to, well, how would you then take this species diversity and create a marine or terrestrial reserve to maximize biodiversity. So we're going to go over some of those techniques on Wednesday, and then we'll reserve Monday, the following Monday, to do a review, and we'll just highlight all the terms that we learned and the techniques we learned, and make sure that we have, like, just a solid footing about what these things mean. I'm not including a reading for this ordination stuff because I'm referencing what I'll get into, which are two different primary sources. One is the hands-on with machine learning and using this initial technique for doing ordination. And the other is this vegan tutorial, which is an R package that is more commonly used in like ecological analysis. So yeah, so hopefully you guys are feeling like, okay, here's what's expected for the lab. Don't stress about the midterm. We're going to have a review, and we're really going to like bulletize all the terms that you should know. The midterm will, it'll be open laptop, but no communication with each other. So, I mean, we'll hopefully be in person, and it's an honor system, but it's not something I just think that you guys should, you'll have your materials in the real world to be able to reference, and you should be able to reference them here too. But you're gonna have to have some familiarity with this stuff before you go into the midterm, but you should have that just by sake of doing the labs. And I will also post an anonymous feedback. I did that last quarter with the project management course, and I'll put that this quarter. I know it's been a little with the remote learning, and not as much text as should be there with the labs. It's been a little squirrely as we get into a rhythm, but yeah, I really appreciate the feedback that you guys have been sharing with me, and encourage more. Okay, so yeah, and then so the species lab is going to be due Wednesday. I've got this reserve, this communities lab due the following Monday, so it wouldn't. Let's see, is that so because it'll be another week since we will have finished this material, and then the reserves will be right now it's scheduled for again for a week after we do that, but it's also a smallish lab, so these are kind of back to back, but you guys have already done the species lab. It's just answering a couple other questions that I peppered in there, and then this communities lab is just a matter of just running it, and I'll be adding a couple more questions with the lab submission and the rubric for that too. Yeah, and really this is giving you exposure to these techniques and understanding machine learning in a broad sense and how it's applied to the environment. But with any one of these topics, you could get, there's entire dissertations and careers made on any one of these things. So it's really meant to give you some initial introduction so that some of the basic terms, and then hopefully when you use it in with your capstone projects or out in the real world, or then you have some starting points with which to apply them. Okay, so I pretty much covered these two items for the announcements, where we've got the lab 1 submission just with the Google form, and then rather than extending that reserve planning module, we're going to dedicate next Wednesday to being a really thorough review. And then just before I move on to actually getting into ordination, any other comments or questions about the course organization or lab submission?
I have a question, Ben, about Lab 2 and the iris data. I know that in previous classes, we've talked about the racist history of that data, specifically with it being posted in eugenics publications originally. Is it possible for that to be changed to the Palmer penguins data, since that was specifically created to replace the iris data?
Yeah, I had no idea about that history until that was emailed to me. And this is it would be pretty insane to suggest that I'm promoting eugenics because of using a data set on a flower properties. So obviously not that, but I'm certainly cognizant of the concerns and with certainly equity and not wanting to promote racist beliefs. So honestly, I mentioned the penguins data set because I just thought they were really cute and I like penguins and marine life. And I can go back and update that because that's a little sorted, but I also just want to take a moment, just to mention something that I think that, this is a longer conversation that we actually have slotted for a reflection period with the whole module on ethics and justice. And we live in a cancel culture that I also find a little bit scary. When there's it's really important that we can differentiate products from people's perspectives. So, I mean, just by using an iris data set, could I be canceled? It's kind of like it's a pretty highly charged and judgmental time, but it's just, anyway, gives me I have some also some perspective about the fact that we have to take the good with the bad, in the sense that it's good to be able to parse out what's useful versus what's not, and just simply eradicating anything in any association with belief systems that are obviously outdated and inappropriate, and all things associated with that. That seems a little. To me, it's a little scary, because it's actually the beginning of the thinking that led to horrors like Hitler Germany. It's this kind of all or nothing maxim that I just would caution you all to think carefully about. So with that caveat, I can bring in the Palmer penguins because I don't want to be canceled. And I think it's important to promote equity and fairness, and we don't need these associations unnecessarily. But I also, it's good to differentiate the dude who had some pretty horrible views from 100 years ago with, a data set that's about the dimensions of a flower. So yeah, I hope I haven't offended anybody, but I just think it's important that we have real conversations about this. And it's not that I have, truth, and I should probably be told what's up in so many ways. And I'm definitely open to that. But I just wanted to caveat that the differentiation there, that I hope we can all, as adults handle. And it's, they also say a sign of intelligence is being able to hold two opposing opinions simultaneously. And these are opposing things. There's like real benefit on one side, and there's ugliness, and there's, our history is fraught with ugliness, and we need to look at it with open eyes and then really assess. So anyway, I hope I haven't offended with that take, but I can swap out iris. I had no idea it was associated with the racist. dude and with the Palmer penguins, and yeah, we can move on. Clarissa, have I offended?
I mean, I just think that when there's an option that works in the same way that is not
Mhm.
connected to white supremacy, that we should definitely use it. And I don't think that that is part of cancel culture. I think that that is part of us recognizing some of the terrible things that have happened in our history and changing what we're using to use something that isn't connected. Like, I don't think that's cancel culture. I think it's us like updating what we're using.
Mhm.
And I think it's really important to do that. And I think it's part of having those adult discussions that you're talking about and recognizing that there's something else that works in the situation just as well and is actually like newer data. You know, it makes sense to me to be updating and using newer data, even if the iris data wasn't racist or didn't have those connections, because that data was collected a long time ago and technology is changing. And I don't know, I don't think it's necessarily part of cancel culture in the way that you feel that it is.
Okay, yeah, that's totally fair. So I agree that if we don't have to have association, there's perfectly reasonable alternatives. Then yeah, we can go there and be supportive of the world we want to inhabit, as opposed to just the way things have historically been.
I think it's especially, yeah, and I think it's especially important given that science has historically been pretty exclusive of non-white people.
Mm-hmm. Yeah, cool. Well, I really appreciate these conversations because it's very much of the moment, and honestly, it heartens me that this matters to you all, because you are the next generation creating a world that hopefully is less polluted, more sustainable, not only environmentally but also socially and inclusive. So yeah, it's really important. So it really does make me glad that you guys have these values moving forward, because yeah, and these these tools, like I had mentioned before, it's very much in the news. There was a former Facebook employee that testified before Congress about how the damaging the social feeds, which are all AI driven. That's all machine learning being applied to like what's going to keep your eyeballs entrained. What's the next news story that will captivate your attention so they could increase their advertisement dollars, and that's based on creating inflammatory content. And that's a known fact with Facebook algorithms, because that's the way that's hacking the brain. And so, if we don't understand how the use of these algorithms can malign society, then we're doomed to repeat that mistake. So anyway, we're going to keep on chewing on this, and I appreciate the feedback. And again, I think it's awesome that you guys have this as a priority. So let's keep on checking and keep on flagging, calling me out whenever I miss that opportunity to be more inclusive. Okay. Any other comments or questions about the overall structure or fairness? Yeah. Cool. Okay, let's talk about ordination now. And okay, so ordination is basically the ability to take multivariate data, like highly dimensional data, and then be able to reduce the dimensionality of it. So previously, you guys did a clustering exercise, and you had this dune data set, and you had a bunch of species in each column, and each row was a different site. and then it was each cell, the intersection of the site and the species says like, what's the percent cover of that species? Well, you can take that and you can actually reduce it to, let's say you have 24 species in the data set you'll be using in lab today. And then you could collapse all that variability between rows or observations or sites and into just a couple axes, or maybe 3 axes. So that's a dimensionality reduction. And it's basically capturing variability in with fewer terms. And that's the idea of ordination. And what it does is, mathematically, there's some terms that I think are less important for you to understand all the heavy math with like eigenvectors and eigenvalues and the linear algebra used to achieve these, but understanding like the properties of the different techniques and understanding that essentially it's taking those observations, so let's say each row, and then it puts them on an axis with each other that then puts similar ones closer to one another, and the dissimilar ones are further away. So that's what that dimensionality reduction is trying to do, is trying to take all that variability across multiple columns and then reduce it into fewer, basically, columns, and then plot it out in an axis. And there's a certain amount of variance of that variability that can be described based on the axis that, let's say, initial 1 or 2 axes. And then you can get more and more variability as you add more axes, more dimensions to explain the original data. But so this is a simplification of the input data, and it can be used in sequence. So this is, for instance, sometimes you might have really complicated multivariate data, and then you want to do something like predict presence. Well, you could take all those environmental variables that we had and then collapse them into these ordination axes, and just use those as your predictors. So these things can be used in combination sequence, and it's not meant to blow up your brain, but just think about them, individually, how it works. And then you could be like, oh, well, maybe I could do that initially and then go back and do another prediction later. So these terms here, PC1 and 2, and then MDS1 and 2, those are generally referring to the axes that we create when doing this ordination, with the different techniques associated. So principal components analysis is probably the most common, and it's been around the longest as far as an ordination technique. But it does also assume a linear relationship between your terms. Non-metric multidimensional analysis, which is certainly a mouthful. It's more flexible, so it can capture variability even when assuming that the correlations between your columns are not linear. And in an ecological setting, you can also do this extra thing where, or they have these extra terms that you'll see in the tutorial, which is unconstrained ordination, which is just talking about species, like you already did with the clustering, but now we're using an ordination technique. And then if you want to actually throw in species and the environment into the ordination, then that's called constrained ordination. So it's basically just using the same tool and expanding upon what it's doing. And I just want to also caveat some of these terms that you'll see in the materials referenced. Yeah. Oh, so I meant to hide that one, and I might go back and update that. Where's the hide? That used to be a hide format. Edit. Huh. Oh, maybe it disappeared as a feature. I'll just move this to extra. Okay. All right, so let's take a look at what does this look like? Well, in the lab.
Sorry to interrupt, Ben. I think you can maybe right click if you want to hide the slide. Sorry, continue.
Yeah, I thought I did that. I was on the slide and then I'm doing a right click, skip slide there. Why didn't it? Maybe I wasn't highlighting the slide. Thank you. That's what I was looking for. Cool. Thank you. All right. Okay, so now we're going to look at these the species data sets, different than the dune, same idea though. In this case, we're looking at reindeer grazing habitat, which is this they call them lichen prairies, and so this is, each site is row wise and each species is column wise, and then we've got some percent cover, I think, are the values in between. So the sites here are the row numbers, and then we've got the species here, and you can see, you know, if it's a tibble, it does that nice display of just showing you column-wise species, and then saying, oh, now we've got 37 more variables besides the one shown here. So those are additional columns down here. And this is common too, to call species by the first four letters of their genus name, and then the first four letters of their species name. And so these are all these species found in the vegetation of this reindeer habitat. Okay, so that's it's a lot of dimensions. So we've got 24 rows or observations or sites, and 44 columns or species. And what we can do is run this an ordination on this data, and then each of these sites then is labeled here with a number, which is, you know, the same as a row. But now we've explained most of the variance based on these two axes. And so there's some residual variance that it's not explaining, but this is the first order approximation of it. And these are the sites listed by row number. And then the species becomes these axes. So it's this first axis and these second axes. It could even be just one axis, because this is just an ordering operation. And then as you add more dimensions, it can basically fit more of that variability. So that's why we call it ordination, is because it's ordering things from most similar to least similar, and by collapsing the variability found within the underlying data. And then we could do this other thing where, let's say we've got these species on this axis that explains that variability, and then you can get these vectors out of, well, how does the environment map out to those sites? So in this case, we're looking at different chemical properties of the soil associated with this. So pH or acidity, aluminum, iron, magnesium, calcium, nitrogen. And each of those dots, remember, this is actually the same as that plot. No, it's not the same as that plot before, but each of the dots is representative of each site. And so you can look at site by site, and this is quite a big picture view of how each site then is spread according to species composition. and then looking at, well, how does the environment overlay? So this is a secondary operation where we did not ordinate based on the environment. We ordinated based on just the species compositions within the sites. And then we secondarily asked, well, how does the environment vary? And the length of this vector corresponds with the strength of that, that gradient. So this is increasing nitrogen as you move towards these sites and increasing acidity as you go towards these sites. So it's directional, and the length of that also indicates how strongly that gradient shows up in that data. You can even go further, and this is kind of wacky because this is actually a generalized additive model superimposed over this surface. So what we're looking at now is if we just separated out aluminum and calcium from this plot, it's the same plot. And then what we can do, so you guys were looking at generalized additive models that allow you to have like a smooth function, but you can actually have that smooth function you can apply to these axes, and then in that case, it's trying to fit contours that we see. One is red. I'd have to look at the code to remember which is which, but and one is green. So calcium is either red or green, and aluminum is the other one. And the point is that you can then look at how that gradient changes across those sites that are spread out by species. So yeah, with really complicated multidimensional data, you need these ways to kind of reduce that dimensionality into something that you can kind of wrap your head around and start describing what the environment is doing and how it's differing across sites. And in this case, you can do it across, you can look at this graph and say like, okay, this is, let's say calcium is green. Well, this site is very different from this site from a species composition perspective because they're far away on this ordination axis. And this is how, and calcium is generally increasing between the two. Whereas, you know, along this contour, they're not really changing much from a calcium gradient perspective, but they are different according to species composition. Otherwise, they would be closer together. So quite a bit of information packed into this single figure that is kind of getting at all these multiple dimensions being collapsed. And so far, again, we're just looking at species composition being reduced into this ordination axis, and then the overlay of the surfaces of the environment. You can also throw in species and the environment into how it differentiates the sites. So now the spread of the sites is based on not only the species, but also the environment. And that's where in ecological terms, they tend to call this constrained ordination. So we're constraining the sites of the observations also based on the environment as well as the species. And so in that case, they've used a slightly different technique, canonical correspondence analysis. And so for the labs, you guys are going to go through and just run this stuff, and I'll add back in some questions to kind of force some evaluation of what you're looking at. And actually, I'm going to do this. I'm going to run this here because I just want to make sure I can install stuff. Maybe I'll do that. I'll do that after.
Hey, Ben.
Mhm.
Do you mind if I jump in with a question?
Please. Oh, please. Yeah.
So I understand that the last graph we were looking at is considered to be like constraining the ecological model based on like environmental variables. Would you mind explaining like what it means to be constrained by the environment and like maybe how we can interpret like that graph and like why like the blue lines in this graph are different than the unconstrained graph in the previous slide?
Mm-hmm. Yeah. So the difference is that the spread of the sites previously was only based on how the species composition differed. And now the spread of the sites, and meaning also the definition of those axes and what they are composed of, is based on the environment. And essentially what it's trying to do is maximize, look at the variability across the sites, not only for species, but also for the environment, and put that into a space, an ordination variable space that then explains the variability seen between sites, not only for the species, but also for the environment. So the spread of these points, like here 13 is at the top, 24 is at the bottom, that's different than here. We see 21 and 14. And so those sites are, you maximize the separable differentiation between the sites by, when including the environment, that's going to have an influence over how that spread gets dictated, and also then how those axes get defined. Yeah. So it's essentially just like adding just more columns of stuff with which to run the ordination and collapse it down. And in this case, this vegan package, it can keep track of what's the species versus what's the environment, because that's so commonly of interest, and because you also would want to differentially see, okay, here's where you can then still add those vectors of what the environment is how that's behaving, versus where the species are that we also see associated. So I didn't show that in this figure here, because it does look a little complicated when you show that. But the spread here is based on the species that are coded in red, but they tend to get overplotted when the figure is this small. But you can see that some of the sites basically are defined by having more of a presence, a higher value of some species than others. And that's how the spread of those sites based on those ordination axes is based on the spread, maximizing the spread of those values of species in red. And this one does both, the constraining ordination by environment. So I think that this will also help to look at some of the underlying materials for this. So in the ordination lab, whoops. We're basically, you guys are going to use this, go through this hands-on machine learning with R chapter to do the simpler, more common technique. It's not using an ecological data set, but you could just imagine swapping in species. And then this one is this vegan tutor vignette. I had to compile it, but you guys can download this and look at this. And so just to go back and revisit that question, what's the difference here? If we look at this vegan tutor package, and then actually in the lab, you're going through these two basic techniques, the principal components analysis as a first whack, and then this non-metric multidimensional scaling analysis. And then you're going to be doing the unconstrained just on the species, then overlaying with the environment, and then constraining on both the species and the environment. And so in this case, I'm suggesting that you read the entire chapter of chapter 17, because it really talks about, well, how many axes do you need to explain how much variance? And I think it does a nice job of kind of walking through what these things mean. And in the vegan package, in this tutorial, it talks about a few extra things that they're less important. So we need to constrain how much you guys chew on at a time. So I focus you in on the specific bits that you need to. So here's non-metric multidimensional scaling. So we're talking about section 2.1 of this PDF. And so that starts here, talking about what this is. And we went over Bray-Curtis dissimilarity last time. So that's how we get the initial distances. And then it's using those distances to try to figure out how to ordinate and create these axes. And it kind of talks about the stress plot. So it's trying to reduce the stress. The stress is where there's not a perfect, where the distances between the points do not equal their distances in the matrix when you try to flatten it to, let's say, 2 axes. So as you expand the axes, basically going out to the number of, let's say we have, what was it, 24 species. Once you get out to 24 axes or dimensions that you allow for ordination, then you would repeat exactly the original data and you would have 0 stress. But as you're trying to reduce the dimensionality, the distances that are actually measured in the output ordination are then different from the original data when you look at the pairwise distances. So the stress is just the difference between that. How close are we? And it's trying to reduce the stress as you iterate through the dimensions that you add. So far in the examples, I showed just 2 dimensions because that's what we could very easily visualize. But you can actually get into like 3 dimensions or many more. It's later on. I think it's when we actually get into the environment. Yeah, with the constrained ordination, there's a 3D plot. So you guys are going to do one of these, and just the notion that that you can have multiple axes, and of course we can't really visualize beyond 3 axes at once, but it's just trying to manage. It's trying to reduce the stress between how we lay things out on those axes, meaning the actual distances measured versus what we can fit into that limited number of dimensions. So I point you to specific sections of this tutorial to give you, more language to understand what this is doing. And then with this other example, you're looking at it's like this checkout. It's kind of fun. It's a data set based on what people have in their grocery basket, and which sounds kind of silly, but if you actually look at this and think about this a little bit more, this talks about how if we look at the items in the basket, there's actually a correlation between them. If somebody's getting cheese, they're also more likely to get mayonnaise. They're thinking about sandwiches. I don't know what Bulmer's is, but I'm thinking it's a beer, and I assume Foster's is. So it's like, this is the beer run. Cheese and bread, lasagna and pizza. So that's what it's using. It's looking at how correlated these are, and then reducing the dimensionality and how you can explain the presence of these things with fewer columns of data. And then this is the mathy perspective on finding these principal components. So a component, an axis, a dimension, I'm using these terms interchangeably, and they get used interchangeably in this literature on ordination. And these components, you're looking for what explains things. So if we have these two features, the first component, and here you do see a vector of how strong that correlation is between these two features. And this first component is going to take care of the variability along this axis. And the second component maybe along this axis. And then a third is going to be some other little wiggle, and you can keep adding components until you have as many data points as are in this data set. But you can think of it also in the principal components, it's just fitting linear planes in the data as you keep on adding data, and that's why it's linear. This lab is using this other framework. We had previously used something called Carrot to help us with doing some of the logistic regression. This is another one called H2O. And it's just a way of setting up your training data set, which method you're going to use. K is like, what's the default number of clusters you're going to have? And in this case, it's the number of columns that are in there. And then impute missing, that's actually filling in any blank data. So this is, in this case, we have many axes. In this, it's getting up to 42. So that's as many columns as we have. And you can see that here's the proportion of variance. So it's 5% initially explained by the first axis, another 5%. So now we're over 10% explained by these two axes. And then as we get to the last axis, then we're like 1%. So it's a declining amount of variability explained with each of these additional axes. And then this is the mathy part, is that it's using these things called eigenvectors to basically apply a vector that transforms the data in a way that can explain the variability. And then you can see this is similar to what we've seen about variable importance, about kind of what is the structure of a given axis, and what are the weights associated with that axis that explain it the most. So in this case, Ballmer's and red wine and Foster's. So maybe there was more beer runs in this data set of grocery baskets. And that is a stronger predictor. And it's a component. This is the component axis, the first component of the PCA, and how to describe that component, what it is comprised of, how it collapses all that data into a single component based on these weights. And then here's that one axis. And so this is where we have more Ballmer's, red wine, and Foster's, Cronenberg on one end of the axis versus the other end, which is maybe the non-alcoholic end. And then this axis would then be described by a different set of your input data and weighting that, probably not on alcohol, maybe like on sandwich goods, that kind of thing. So then there's a question of how do you find, how many dimensions do you need to explain the data? and you can apply several different criteria for it. So if you know what percentage of the variability you need to achieve, then you would just add up the proportion of variance until you get to how much you need explained. Or maybe you know that you need the simplest first cut and just 2 dimensions, or maybe 3 dimensions. And then you can report out how much variance you're explaining with those dimensions. Or you can take this other approach, which does a cutoff. So this is looking at the elbow, and where that value like starts to taper is considered the elbow, and you'd use this value here. So this is 8 dimensions you would use before it starts to just have a non-significant explanation of variance. And then this is a more explicit approach here. So yeah, this is, I mean, it's there's a lot going on with this, obviously. It's kind of a complicated topic, but hopefully working through some of the data sets and the code will help explain how this works. I would also say there's, this is, gosh, the non-metric multidimensional scaling is actually kind of complicated. It's kind of like random forest, how it's not just a recursive partitioning tree, it's a tree of trees. And in this case, it's a whole bunch of ordinations, because there's this heuristic, this random component about the initial conditions. And so it handles all that randomization and then the reweighting for you. And this is what it's doing. And there's this aspect also of the fact that if you just rotate, depending on where you set the initial axes up, you can get different results based on how you rotate it. And that's this thing called procrustes, to rotate the axes to look at how that might influence the values. And it basically does that for you. And so we could go into, all the gnarly weeds of each of these techniques, or just basically understand that, yeah, these are the basic idea is to reduce the ordination. This one does it flexibly, but there's some extra stuff under the hood it needs to do to handle that flexibility, including running a whole bunch of these and then weighting the results of those in your final output. So yeah, I hope you have fun with this, and I'll add some extra questions into the lab for you to include in the lab write-up, and I'll include that like the rubric for the grading. So yeah, and maybe I'll field questions now, but you guys should be able to. I think what I'm going to do is just make sure I get the packages installed that you need on Taylor, and then be available for the next half hour for more questions, and we'll just leave it open in this format. So if you guys want to stick around, you can. Otherwise, no worries if you want to do other things.
There's some questions in the chat. If you could.
Thanks.
Thanks.
Thank you. Yeah. And I guess I'm wondering, I should have also had the Slack going too. Maybe there's questions over there, because then you can also have them for future reference. So maybe I can copy paste in the Slack if we want to have it. Ronald Fisher, this guy, so he's super sucked, huh? Yeah. Okay, I need to do my due diligence there. Make sure I don't repeat, promulgate works that promote these opinions. So thanks for chiming in, Sharad. Are there instances in which you'd want the unconstrained ordination over the constrained ordination? Or is the constrained ordination with the species and environment what you'd always want to use, given you have that environmental information? Okay, that's a great question. Yeah, so yeah, why, if you have the information, why wouldn't you just use that? They are telling two different things. And this, what you'll see in some of these outputs with the ordination, like we were looking at that percent variance explained, it might not, adding the environment might not really explain a lot of variance. Meaning here we see K, which is potassium, and P is phosphorus, and Al aluminum. So if you look at this axis and you did the constrained ordination to include species and environment, you might conclude that, wow, well, okay, so this is really, these sites and the species composition within them is really determined by potassium and phosphorus. I mean, that kind of, you know, we understand that like the rate limiting nutrient in for a lot of plant species, for instance, is phosphorus and nitrogen. So that kind of plays out. But what if the variance explained on that ordination is only like 5%? Then this is not actually that informative about how these sites are distributed. So it's kind of it's another way to think about the ordination is really this is a crude approximation for visual interpretation of multivariate data between observations. So it could be might not explain a lot of variance. The question might be different too. So, you know, maybe more fundamentally, you're interested in how the species are distributed, and then you want to secondarily ask, well, what are the environmental trends in it? And that's what that overlay does. It's actually not ordinating on the environment, just the species. And so it is that secondary question. But you could just also flip it, go the other way and just ordinate just on the environment. And then look at how that spreads out. And maybe that would determine, for instance, like you might want to do that. Let's say you're planning on conducting a survey of reindeer, and this is, reindeer foraging habitat. And you might want to see like, well, what do they eat? Well, I don't know what's on site, but I do have environmental soil information, digital elevation models, and other things I can remotely detect, heat, exposure to sun, precipitation, etc. So maybe what I'll do is just ordinate on the environmental data, and then create a survey design that maximizes the sampling of that environmental space. So that would be like, that is an approach commonly done for survey design, for actually understanding, like going out and counting critters and figuring out, like making sure you're getting an environmental spread on where you go to look. Other scenarios, yeah, I guess it really just does depend on the question that you're asking, how much variance gets explained. You may or may not include it depending on how much variance gets explained. So yeah, it's a great question. Any other comments about that, or maybe do other people? Has anybody else come across ordination data, especially in the environment? Okay, that's fine. It's new. It's definitely a lot of stuff to jam into basically a very limited plane or space of these axes. And actually, I learned about this doing grad school in a multivariate analysis class where we spent a lot more time on it with this really awesome guy, Dean Urban, who's written a lot of awesome papers on this topic. The header title should be TV. Oh, thank you. I'll fix that. So if NMDS is flexible, could it also be applied to linear relationships? Definitely. Yeah. And I guess if you know that it's linear, that you might want to choose a simpler technique. But even knowing if it's linear, you kind of would need to look at each of the variables with each other, each of your columns, kind of like a pairs plot. Look at them with each other to see what that relationship looks like, if there is one to be had. But yeah, so I would say now in ecology, the default tends to just be NMDS because it can account for these other factors. Before there was this extra, it's a meta MDS is what the function is that you're using in the package, our package vegan. And that's what handles the changing the initial conditions, rerunning things and reweighting things. Before that was in place, people shied away from using NMDS because you can get a fairly different result depending on when you ran it. You just run it again, you get a different result. So that was so I would say with that innovation, with the meta MDS function in vegan, it's now the most commonly used technique in ecology. But in the broader like machine learning community, I would say PCA is the most commonly first used technique, and there are others that that get kind of a lot fancier in this dimension reduction section. Generalized low rank. This field just could go on ad nauseum, and it's kind of like I just want to give you guys the simplest, most common in the general world, and then the most commonly and sophisticated technique used in the sort of environment and ecology world. And we'll just kind of like leave it at that. But just know that there's a lot of other ordination techniques out there. And like this generalized low rank models, you can kind of tell by the name, like it's ranking. So it's that, again, that's just part of that ordering process. It's where you get the word ordination. It's just trying to figure out how do you order each of your observations in a way that explains the amount of variance you have and however much data you're trying to throw at it. Yeah. Okay. Okay, and then CCA. So again, that vegan tutor package does talk about the difference, and it's just like some of this stuff is just kind of nauseating with all of its history. Like the author, it was called canonical correspondence analysis, but then if you look up the word canonical, it's like he bickers with that term being used. So anyway, it's just another technique that does a slightly better job than PCA with linearity, and it's sort of, but it's not as robust and or complex as NMDS. And when throwing in the species and the environment, that's the most sophisticated technique that the vegan package offers. So that's the last example. Yeah, it's NMDS is not applicable in that constrained sense with the environment and the species, just the CCA is offered. Okay, oh, kelp forest ecology. Yeah, cool. Yeah, I love kelp forests, so I love that stuff. I was actually just, I got out to Santa Cruz Island yesterday and highly recommend you guys hopping on Island Packers for $30 and spending a day hiking out there. It's gorgeous, and the kelp is blooming, and I see Colin's got a kelp background in the background, and even the name for kelp is just the coolest thing ever. Macrocystis porifera. So it's like a big ball of gas that causes the leaves to float. The macro, the big cyst ball, and porifera because it looks like fire with the blades going up. And it's the fastest growing plant in the world, foot a day. Fun facts for the day. Yeah, let's see. Do we have anything else in Slack? And is there anything in Slack that I haven't addressed that you guys would like me to talk about here? I'm going the wrong way. Oops. Whoa. Go to the top. What's going on here? 14 days ago. This is so weird. Am I not understanding how threads work here? Ah, there we go. Oh, cool. Ah, look at that. Oh, that's so awesome, Colin. Thanks for sharing. NMDS in Actione. Cool. This is great. Colin, do you want to share anything more about this?
Yeah, it was for like a community trend report examining like differences for like 24 MPA reference sites. So the big takeaway from this one is just that Santa Rosa is kind of a colder water island, and so it kind of sticks out to the left. That's like the purple one. And then you get this big like El Nino in like 2015, 2016, and kind of 2014, it's a little bit warm as well. And so it kind of starts coming closer to the other warm water islands, which is kind of interesting. So you can kind of see them like merge in a little bit. It's kind of interesting. It's really low resolution, I think, but the original is a little bit easier to read.
Wow, this is so cool. I love it. Oh, excuse me, sorry. So Colin, are each of the points here a different site and they're color coded by island? Is that correct?
Yeah, they're color coded by island, and then the points are either circles or triangles for whether they're inside a reserve or outside. And you can see that the communities are very similar, whether they're inside or outside a reserve, if they're on the same island. It's hard to see in this plot, but there is an old reserve, Landing Cove and Cathedral Cove, are sites on Anacapa, and they tend to be kind of out by themselves because they are much different from the rest of the sites at Anacapa. You can also do this in 3 dimensions, which I have on like a Shiny app. And there you can see Landing Cove and Cathedral Cove are consistently just like way out by themselves, like with nothing else, because they're so different. But,
wow, awesome. Yeah, feel free to share the Shiny app if that's available online. Yeah, that's so cool.
Yeah, I can put
and
that
just
in Slack.
and also, Colin, just to confirm, like the relationship with what we saw before, did you feed a data frame of sites row-wise and species column-wise with maybe some measure of count or percent cover?
Yeah, exactly. So this is with, I think, about 75 species, and it's all count data. So there's about 75 columns for that, and then there's each row is a site, and then it's like looped. It's like grouped by year as well. So it's like grouped by site and year, and then they calculate. You use this ordination ellipse also to get the like 95% confidence interval around it, and that's a bit weird, but
yeah, that was one of the sections I didn't really go into here. But
yeah, it's hard. It's
the
harder
Ordi
to
ellipse.
do.
It's in that. Oh, that's not the one I was referencing. The vegan tutor. Yeah, here's the Ordi ellipse function. So it is in that little tutorial. But wow, that's so cool. What a great example. I gotta add this to materials. So yeah, really, really cool. If you want to share that, the full reference too, Colin, that'd be awesome.
Yeah,
So
definitely.
great. Yeah, very cool. Any other comments, Colin, about your experience doing this stuff? Was this helpful? Is it getting picked up in management or elsewhere?
Yeah, it's kind of a small part of the larger report, which also uses random forest, which is kind of cool to like kind of rank species importance for reserve status. But it's not published yet because the Park Service is super slow about stuff like that.
Yeah.
But yeah, we do like community diversity and then community similarity, and then like ranking species with random forest. And then we use GAMs to model the species that are most important for predicting reserve status. So it's like, okay, here's the list of species we want to look at. We do GAMs over like the species modeled by like the island, the reserve status, and survey year, and the relationship between island and reserve status to like see like really what are the driving factors for those species, either biomass or count, things like that. And then we also do like trophic relationships where it'd be like kelp predicted by like sea urchin biomass and lobster and sheephead and those sorts of things. So you can kind of see like what species are affecting the biomass of certain species.
Very cool. Oh, that's so awesome. I love the real world example. So like these techniques also, GAM, random forest, NMDS, like you guys are actually, you're doing only the cursory, touch base on them, but it's really cool to see the real world stuff being applied and right here in our backyard with the National Park Service collected survey data. So yeah, awesome, Colin. Thank you so much for sharing in there. Had you been working with them before you started the program then?
Yeah, I did 5 years with the Kelp Forest Monitoring Program. As a side note, when I started learning R, I was actually using your code that you had wrote for that same program for our annual reports. So that was what I based my learning of R off of.
Oh, funny. Yeah, I need to touch base with those guys again. I was supposed to do an R workshop for the National Park Service, and then COVID happened and it just like ding ding bounced out. So but I'm glad to hear that they're still using that stuff, and yeah, super cool. Wow, and this is way more advanced than whatever little plotting things I was doing. So yeah, this is super cool. Love it. All right. Well, next year I'll have to have you back, Colin, to as a guest lecturer, share on this. Very cool. All right. Well, anybody else have? I mean, you can't follow that. Yeah, there's nothing to. Mic drop. That's awesome. Yeah. So, really cool. Let's see. Any other comments? Well, I'm going to make sure that that everything's installed on Taylor with the packages. So, and then I'm just going to kick around if you guys have any other specific bugaboos. Last time Grace had some issue with the library paths and got sorted there. If you feel free, I'm around for the next 8 minutes. Colin, do you have designs to go back and continue working with the National Park Service, or you haven't gotten that far?
Well, David Kushner retires in October of this year. And
Oh, wow.
so that position's opening up, as well as Josh Sprague, who is the lead for that program. He just moved on to Crater Lake National Park. So his position is currently open, and they haven't posted the job listing for it yet. But so I'm hoping that I'll be able to, I mean, I'm going to apply for sure. There's probably a lot of super qualified people going to apply for that one though.
Wow, awesome. Yeah. And did you get to actually go diving and do some of the survey work? Oh man.
Yeah.
Yeah. That's so cool. There was this conservation GIS conference I went to years back where this guy described this very strange dualistic existence where he'd spend, half his year out in the field in nature and just and then this is back in the early days of green chromatic screens, just in a dungeon coding. And he's like, it's hard to reconcile these existences. And I'd say the one bummer about doing more of the compute stuff is just missing the original inspiration of being in the field. I had done as an undergrad at research diving out for the Gaines Lab in out on the islands from Baja all the way to Monterey. And it's definitely why I love this stuff. It's the love of that encounter. Yeah. So to be able to work it, so you actually do analysis and then you get time in the field, it's kind of the dream.
Yeah, part of the whole reason I did the trend report was because COVID, it killed our field season. So we had 6 months to just work on stuff. And I was working with a guy who used to teach statistics at Humboldt State. And so he was pretty good at directing the ecological statistics we needed to do. And then I was pretty good at the R stuff. So we made a good team doing that.
Awesome. Wow, that's great.
Hey, Ben.
And then, yeah, please.
So I was just going on to Taylor to try to retrieve the path for submission for Lab 1. So you're saying that it's supposed to start with users and end with the RMD. So when you do print working directory in the terminal, you only get the second half of the path because it starts in your user already. Is there an easy command to run in the terminal to get the whole path, or should I manually piece it together? I'm just a little bit unfamiliar with anything besides PWD.
Yeah, so you can get working directory. Another thing that you can do, it's a good question, is R's got all these little tricks. So as if I'm here under let's see, docs, say what you can do is you can start typing DO and then I can hit tab. Oh, doesn't do it there. If I'm in an R markdown, you can usually start a quote string. Weird. Oh, you know what? Because I'm not actually in a project. I think that makes a difference. So if I was, let me log out because I finished installing. Sign out, I'll sign in. And if you have an RStudio project, I think that's kind of the difference. Yeah, cool.
Wouldn't mind just running through how you just easily pull the path all in one move? That would be awesome. But if it's not possible, that's okay too, because I can always retrieve the beginning and piece them together.
Yeah, well, if I'm in a project, let's see. Sorry, once I'm in a project, I'll show you what I mean. Wow. Wait.
So the project sets the working directory. Can you hear me? My earbuds, I think, just knocked out.
I can hear you.
Okay, cool. So yeah, the project, opening up the project sets the working directory. And then if I'm in, I think if I could say ls or list.files, yes, and it would list the files, but that's kind of clunky. So what we can do is if I'm in this and I've got, let's say, data, SDM. So if I want to, I can usually hit tab on a quoted string if I'm in a project, and it will automatically provide paths, and then I can tab to select things and use up arrow and down arrow. So this would work if I started with the users. And actually I don't have a user, but Julie, are you, there we go.
Yeah, I'm JS Cohen, right? Yeah.
And then maybe it's, I don't know where it's in here, but you get the idea that you can just auto tab complete.
Okay.
So, and select. So if you, any string, R will provide auto completion to any available path. But,
Okay, so you just have to start with that /users and then I can like go from there.
yeah, exactly.
Okay, cool, thank you.
You still might see the screen, right, Joe?
Yes, I can see your screen.
Yeah,
Thank you.
this little drop down and that, yeah. Mhm.
So you just want the file path to the RMD.
Yeah, yeah.
Okay.
But yeah, please make sure that it can run and that you should be able to successfully knitted it. So once I have that RMD, then I can also just look at the HTML. That should be in the same folder. And yeah, and then the rest of the little questions and things that you might want to pepper in there are listed here. So there are the outputs, and then there are questions. Yeah, and then you just submit the form. Yeah. Lots of stuff. Okay, well, I'm gonna go now, but thanks guys so much. And Colin, super stoked to see that excellent example. Feel free to send more stuff my way. I'd love to highlight more products that you guys are actually involved in the class. So cool. Yeah.
I think the repo's in the Slack, so you can check out the app and stuff. So
What? Cool. I'm looking forward to that. Awesome. Thanks.
yeah, see ya.
Yeah.